Nucleus, the administration unit of cell. Every cell gives its optimum or is functioning at its optimum because of the coordinated and the uh, complete functioning of all the organelles within the cell. Not just the organelles, but also uh, cytoplasm and its various molecules play a very important role in the functioning of the cell per se. In this session, we are going to focus on the nucleus, which is considered to be the administrative unit of the cell. So let us look at the learning outcomes of this session. The nucleus is considered to be the administrative unit of the cell, as the blueprint of the cell is stored within it in the form of chromatin, with regulated gene expression to form proteins that are required by the entire cell. So it is the proteins that are the executives, but the information present for these executives to be produced is within the nucleus in the form of chromatin. The nucleus is surrounded by a complex nuclear envelope, which not only is a barrier, but it also monitors the exchange between cytoplasm and nucleus. One of the major differences in eukaryotes and prokaryotes is the fact that the DNA is protected by a nuclear envelope in the eukaryotes, whereas in the case of prokaryotes, the DNA does not have, or the nucleoid does not have, a envelope around it. The nucleus has a nucleoplasm just like the cell has a cytoplasm. Nucleoplasm is a it also has dense nucleolus where the ribosomes are assembled, and a nuclear matrix, which is a meshwork of proteins like the cytoskeleton is for the cell. All molecular mechanisms related to DNA and RNA are carried out within the nucleus with a high level of accuracy and regulation. So when you look at the overall structure of nucleus, what is clearly observed is that the nucleus has a double membrane structure, so that it has an envelope and but it has two membranes. So you have what is called as the outer membrane and the inner membrane, and within the nucleus is present the chromatin, all distributed within and is existing in the form of heterochromatin and euchromatin. Heterochromatin is a more condensed part of DNA, while euchromatin is the less, but the point is that the DNA exists in the form of chromatin. It has the nucleolus, so you can see over here there are two nucleoli present, and the nucleolus is always a dense region because it is a region where you have accumulation of a lot of DNA, RNA, and proteins, and therefore that area becomes highly stained. Plus, what is very clear is that the nucleus has nucleoplasm, just like cytoplasm. So it has a gel-like consistency, and there are nuclear matrix. That means there are a lot of proteins which form a kind of meshwork within the nucleus, and that is what is called as a nuclear matrix. So this is an overall picture of the structure of nucleus. Markedly, what has to be kept in mind is that the nucleus has an envelope which is double membrane. So it's not a single membrane, but it has double membrane present. So this is one of the organelles that has a double membrane. The other two, which are very important in various different cell types, one is mitochondria, and the other is, of course, the chloroplast. So as you can see here, this electron microscope picture very clearly shows that you have the outer membrane, you have one membrane and the second membrane here, and within this nuclear membrane at certain regions, as you can see over here, are certain complexes that are called as nuclear pore complexes, also known as porins. And it is interesting to note that there are more than several thousand porins present on the nuclear membrane. So as you can see, this is a schematic diagram of how the nuclear membrane looks like. So this is the outer membrane. So this is outer nuclear membrane. This is the inner nuclear membrane. And the places where you have a nuclear pore complex, so this entire complex that you see over here is what is called as a nuclear pore complex. Those portions where you have the nuclear pore complex, you will find that the outer nuclear membrane and the inner nuclear membrane fuse at this region. Otherwise, they are two distinct membranous structures. What has to be kept in mind with respect to the nuclear membrane is that just below the inner side of the nuclear membrane, as you can see over here, is present a layer of intermediate filaments called the nuclear lamin. So it forms a nuclear lamin. Basically, you can understand that it is laminating the inner surface of the nuclear membrane. So it is giving a kind of structural support to the nuclear membrane, and therefore the nuclear lamins are very important constituent of the nucleus. So nuclear lamins are made up of three lamins: lamin A, lamin B, and lamin C. And together lamin A, B, and C, as you can see, they are basically forming a base for the entire nuclear membrane. What is also important to note is that the nuclear membrane also is a scaffold for several different lamin associated proteins. So you can see over here you have lamin associated protein 2 beta, you have LEM2, you have emerin, you have the Sun-Cash interaction. All of these are lamin associated proteins and also part of the nuclear membrane itself. Now, many of these lamin associated proteins help the nuclear membrane associate with constituents which are present outside the nuclear membrane and constituents which are present inside the nuclear membrane. For example, you can very clearly see that the lamin at the inner surface is associated with the DNA. Okay, and the DNA at this in this area is heterochromatin. That means it is a highly condensed form of chromatin that is present interacting with the lamins of the nuclear membrane. Secondly, you have certain nuclear membrane proteins that are able to interact with the cytoskeleton on the outside and the nuclear matrix in the inside, thereby establishing what is considered to be a link between the outer environment of the nucleus and the inner environment of the nucleus. So you can have inside out signaling and you can also have outside in signaling. Plus, it also defines the fact that the nucleus in the cell has a place of its own, as the cytoskeleton decides the position of the nucleus per se. And of course, the structure and the shape of the nucleus itself is decided by various nuclear matrix which are present inside, and also of course the nuclear lamina. So in this picture also, what is very clearly observed is that you have the outer nuclear membrane, you have the inner nuclear membrane. Where it is fusing, you have the nuclear pore complexes. These are the regions which allow transport of molecules across the nucleus, and it is very important. It plays a very regulated transport mechanism. So we are going to look at it in a while. But also what is important is that you have the outer nuclear membrane continuous with the endoplasmic reticulum. So the endoplasmic reticulum kind of originates from the outer membrane of the nucleus. And so this is actually trying to tell one important facet of the cell, and that is that the cell has something that is called as endomembrane network. And nucleus is therefore one of the organelles that becomes part of the endomembrane network. As the word suggests, endo means inside, membrane means membranous structure. So within the cytoplasm, you have several organelles that are membranous sacs. So for example, endoplasmic reticulum is a membranous sac. Golgi is a membranous membranous organelle. Lysosomes, peroxisomes, the plasma membrane itself, all of these fall under what is called as endoplasmic network, endo membrane network. And the reason why it is called as network is because each of these organelles are connected to each other. That is, there is a give and take between these organelles, and that is why it is called as network. So nucleus definitely, because it gives rise to the endoplasmic reticulum, is part of the endomembrane network. What is also interesting to note is that on the outer membrane on the outer side of the outer membrane of the nucleus, you can have ribosomes attached to it. So basically, where the rough endoplasmic reticulum has ribosomes attached, the outer membrane of the nucleus can also have ribosomes attached on its surface. So this was the general idea of the nuclear envelope. Now we go into a little detailing on the nuclear pore complex itself. So as mentioned, nuclear pore complex is a huge mega Dalton complex. That means it is made up of several different proteins, and interestingly, there are 30 different proteins present which form the entire nuclear pore complex, and these are what are called as the nucleoporins because they form part of the porin itself. They are called as the nucleoporins. So these 30 different polypeptides are also called as NUPs. 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 NUPs basically meaning nucleoporins. Now, it has a very elaborate architecture and a very defined architecture. Although the structure is not symmetric, it is a highly asymmetric structure, but within that, again, you have certain symmetry also present. Now, for example, let's consider that the nuclear pore complex has two rings. One is present on the outer side of the nucleus, that ring is what is called as the cytoplasmic ring, but you also have a similar ring uh, inside the inner membrane of the nuclear envelope, and that is what is called as the inner ring coat. So you have the outer ring coat and you have the inner ring coat. Okay, sorry, you have the outer ring coat. So this is present on the cytoplasmic side, this is present on the nucleus side, and these that you see, the orange ones, are also ring structure, but they are part of the inner ring coat. And then of course you have the inner ring, so you can see this green, blue and the pink together forming what is called as rings, but these are present within the porin, so they are called as inner rings. And you have what is called as at this portion something that is called as diffusion barrier, and this is actually the one that is forming, this is the pore region within the nucleoporin, this is actually the pore region. So, before we come into this, get into a little detailing on this, another important fact is that you have certain cytoplasmic filaments that are present, and similarly, you have nuclear filaments also present, and this forms a kind of basket inside. So this is therefore the basic complex that is made up of the 30 nucleoporins, and we will now slowly gradually look at the function of the nuclear pore complex. So you must understand that this pore that is present is actually allowing passive diffusion of molecules which are smaller than 40 kilodaltons. But any molecule which is greater than 40 kilodalton, those cannot easily traverse through the porins. The reason for that is because this diffusion barrier that you see contains proteins, so nucleoporins, and within the nucleoporins that there are certain domains present. So we all know that proteins fold to form a tertiary structure. And the tertiary structure in itself can have several motifs and domains. So the nucleoporins have a domain which is called as the FG domains. The FG over here stands for phenylalanine and glycine. So that domain has a stretch of amino acid which is nothing but a repeat of FG. So you have FG, 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 FG present in a particular domain, and so they are called as the FG domains. Now, interestingly, these FG domains make the inner pore quite hydrophobic. Because they make the environment hydrophobic, any hydrophilic macromolecule would not easily pass through. So there will always be a barrier for the movement of something that is hydrophilic. So if you have ions, it cannot move easily. If you have macromolecules that are hydrophilic, will not move easily. Which means that we are looking at the fact that the nucleoporin definitely allows a lot of proteins to enter or exit from the nucleus, and it will also allow RNA etc., to come in and out of the nuclear. But then that all these movements are regulated movements. Okay, so this is something that has been found, and it is understood that this regulation is being done by several proteins, but one of the proteins that plays an important role in the regulation is what is called as RAN proteins. And these RAN proteins are GTP binding proteins. That means they can be bound to GTP. And the fact that it is bound to GTP allows it to regulate the entire process. So we'll just look at it in a while. So effectively, what we understand is that import, that means something that is taken in by the nucleus, and export, something that is pushed out from the nucleus, is a regulated process. There is a traffic across the nuclear membrane, and that traffic is regulated, which is what is very clearly understood, and the nuclear pore complex has a major role to play in that. So what kind of an import export are we looking at? So for example, within the nucleus, you have DNA replication happening. That means the DNA can double, the DNA can replicate to form more than one copy of itself. And that replication process requires a protein machinery. So from where do you get these proteins? Because in eukaryotes, we know that translation happens in the cytoplasm, because all the ribosomes that carry out the the translation are present in the cytosol. So therefore those proteins are formed in the cytosol, but they have to be imported by the nucleus in order that you have a particular function being carried out. Now please remember, replication is not the only process that happens inside the nucleus. You have replication, you have transcription, you have post-transcriptional modification, you have a condensation of DNA that happens inside. So there are a lot of functions that require proteins, and these proteins have to be imported from the cytoplasm into the nucleus, and that happens through the nuclear pore complex. So DNA polymerases, RNA polymerases enter the nucleus. DNA polymerase is for replication and RNA polymerase is for transcription. snRNAs, these are a special type of RNA molecules which are required for mRNA processing, that means post-transcriptional modification. So, we know that 5' capping, 3' A, poly A tailing, splicing, the removal of introns, these all fall under what is called as mRNA processing. And these mRNA processes require snRNA. Now you would say, why do you need the snRNA to be exported out? Because the snRNA has to bind to certain proteins to form SNRPs, or small nucleoribonucleoproteins, and therefore that has to happen in the cytoplasm. So they form the SNRPs in the cytoplasm, and then they are imported back again to carry out the RNA processing. So you can see how certain things have to go out of the nucleus and then they have to come back into the nucleus as well. So you have an import and an export both happening across the nuclear pore complex. So the proteins that are also required for the ribosome subunits to be formed. Please do understand that the ribosomal RNA is formed inside the nucleus, and those ribosomal RNA have to bind to proteins to form the small subunit of the ribosome and the large subunit of the ribosome. So assembly of the ribosome happens inside the nucleus, and therefore all the proteins that are required for the ribosome formation have to be imported by the nucleus. Steroid hormones, for example, they are able to perpetrate their signal by binding directly to a gene in the DNA. And so the steroid hormone has to enter the nucleus to get access to the region where it has to go and bind and activate the gene or repress the gene. So that is also a kind of import that the nucleus has to carry out. So these are just certain examples that are trying to tell you mm, what are the molecules that need to go out of the nucleus, what are the molecules that need to come into the nucleus as well. Now, therefore, the next question that we have to ask is, how does the nuclear pore complex decide what it has to import and what it has to export? That means, how does the nuclear pore complex know that a particular protein has to be brought in? or a particular protein has to be sent out. So a lot of studies have been carried out, and it has been found that there is a mechanism, there is a regulated mechanism which allows the traffic across the nuclear pore complex to take place. So one of the first things that was understood is that a cargo protein, whether it has to be imported or whether it has to be exported, the cargo protein has at its C terminal, at its C terminus, so we know that proteins have an N terminus and have a C terminus. So in these cargo proteins at the C terminus, there are a stretch of amino acids. Mainly they are basic amino acids, but not necessarily so, yet they are higher percentages of basic amino acids in this stretch of amino acids. And this stretch of amino acids is what is called as nuclear localizing signal, that is NLS, nuclear localizing signal. So if you have a stretch of amino acids with a set conserved set of amino acids which are basic in nature, then that cargo protein has to be imported. So you basically understand that the address that is there for the protein to get into the nucleus is present in the protein itself. So the NLS signal is actually telling, hello, I have to be taken in. Okay. And so that NLS signal is something that the nucleoporein machinery has to recognize to allow the protein in. So what has been observed is that across the nuclear pore complex, there are certain mobile transport proteins that carry the cargo with them. Okay, and so on further studying, they found that there are two nuclear pore sorry nuclear transport mobile transport proteins. Those proteins that are importing, when you say importing, that means from the cytoplasm into the nucleus, those proteins are called as importin alpha and importin beta. So in the cytosol, they exist as individual components, but when you have the important alpha recognizing the NLS sequence of the cargo protein, so you have the important alpha binding to the cargo. The moment the important alpha binds to the cargo, the important beta will now bind to the important alpha. Interestingly, now this complex that is formed, that is important cargo complex that is formed, that finds an affinity to bind to the cytoplasmic filaments of the nuclear pore complex. So we know that the outer ring of the nuclear pore complex has extensions in form of cytoplasmic filaments. So those cytoplasmic filaments will now be able to bind to the important cargo protein, and as soon as they bind to this cytoplasmic filament, the cytoplasmic filament will bend towards the diffusion barrier that is there. So you know that the diffusion barrier has a lot of FG domains, okay, because it has the FG nubs. So these FG domains, when they bind to the important cargo protein, the FG domains change its conformation, and as it changes its conformation, the entire important and the cargo protein is now pushed into the nucleus through the basket. Okay, so you can see how the mobile transport proteins recognize the cargo protein. How do they recognize the cargo protein? By binding to the nuclear localizing signal. Once they bind to the nuclear localizing signal, they are now transported across inside to the nucleoplasm. Now, once you have the cargo protein with the importance within the nucleus, inside the nucleus, there is the RAN-GTP protein. This RAN-GTP is always kept as RAN-GTP because of certain other proteins that are present in the nucleus. So what happens is that the favorable condition is that the GEF is nucleotide exchange factors. They always ensure that the RAN protein is bound to RAN-GTP. So the concentration of RAN-GTP inside the nucleus is always high. Okay, so the RAN-GTP can bind to the important beta. You can see how it is binding to the important beta. Okay, and when it binds to the important beta, the RAN-GTP the RAN GTP on binding to the important beta, the important beta loses its affinity to bind to important alpha. And so in the process, what happens is the cargo protein is released into the nucleus. This is what we want eventually. And the important alpha is retained within, but the RAN GTP carries out from the nuclear pore complex into the cytoplasm the important beta again. Okay, now once it enters the cytosol, the RAN GTP is converted into RAN GDP. Okay, so there is hydrolysis of the GTP happening in the cytoplasm, because of which RAN GTP gets converted to RAN GDP. And in the form of RAN GDP, it does not have affinity to bind to important beta. So it releases the important beta, and this RAN GTP again gets into the nucleus. Once it gets into the nucleus, we already know that there are nucleotide exchange factors which it immediately will make RAN GTP into RAN GTP, and so the concentration of RAN GTP is always high within the nucleus. So you can see how across the nucleus, between the cytoplasm and the nucleoplasm, you will always have a concentration gradient of RAN GTP. And that concentration gradient of RAN GTP is what is driving the cargo proteins into the nucleus. Now, interestingly, what has been observed is that the alpha important that is there can bind to another protein which is called as exportin. Now, this exportin can bind to whatever molecules present inside the cell that has to be exported out inside the nucleus, that has to be exported out into the cytoplasm. So those molecules that have to be transported out of the nucleus have what is called as a nuclear export signal. Just like those that needed to be imported have a nuclear localizing signal, similarly those constituents that have to be exported out have what is called as nuclear export signal. And so the important alpha will bind to that and it will go out into the cytosol. So that is how you have import and export happening across the nuclear membrane. So just to go through once again, NLS is a stretch of basic amino acids at the C terminus of a cargo protein, which is recognized by importance. And if you have NES, then it is recognized by exportins. And importance and exportins are the mobile transport proteins, which are regulated by RAN GTP, and they have a concentration gradient across the nuclear membrane. As mentioned, you have RAN GTP concentration always higher inside the nucleus than outside the nucleus. So that is how the movement of the cargo across the nuclear pore complex is regulated. Now, please do understand that there are many other proteins taking part in this, but what we have tried to look at is the one which is majorly recruited for importing and exporting of the protein. Now, another thing that happens in the nucleus is that the DNA gets condensed to form chromatin. You must understand that a very long DNA, to be accommodated within a few nanometer size nucleus, the DNA has to become compact. Okay, and so when the DNA becomes compact, it can become very highly condensed. So the very highly condensed regions are what are called as heterochromatin, and the less condensed regions are what are called as euchromatin. It is very obvious that if the DNA is very highly condensed, okay, then those regions of the DNA are not generally expressed. Now, what do we mean by expressed? That means a G is transcribed by the RNA pore to form an RNA. That is what is expression. Okay, so if the DNA is very highly condensed, then its accessibility to the RNA pore will be less, so it will not be expressed. But if the DNA is less condensed, then its accessibility to the RNA polymerases will be greater, and therefore it can be expressed. So you must therefore remember that to what extent the DNA is condensed decides how much of gene expression is going to happen. Okay, so if the DNA is as condensed as the chromosome at the metaphase, then you must understand that none of the genes is going to get expressed. Why? Because the DNA is in its most highly condensed state. And in that highly condensed state, you will not have the DNA getting accessibility to these proteins because the DNA itself is protected by proteins that are condensing it. So that functionality of the DNA getting condensed is what we are going to look at now. And chromatin is the least condensed form of the DNA. So let us look at what is the chromatin, or what is also called as the 11 nanometer fiber. So if you have a long DNA, the DNA is found to be, kind of wrapped around certain units which are called as nucleosomes. So these nucleosomes that you see are nothing but they are a histone octamers. So they are complexes made up of histones. We all know that histones are basic proteins. Why are they basic proteins? Because the percentage of basic amino acids in these histones are very high. Secondly, what has been also observed is that these histones have what is called as histone tails, and the histone tails are particularly basic. So therefore, because the tails are basic, they have an attraction for the negatively charged DNA, because we know that the sugar phosphate backbone of the DNA makes the DNA overall negative. So this is basically a kind of interaction that is there because of opposite charges. So you have the nucleosome, which is positive in charge, and you have the DNA, which is negative in charge, and therefore the DNA and the negative interact. And in the process of this interaction, what is clear is that the DNA wraps around a nucleosome. So therefore, if you have several nucleosomes present, this entire DNA at portions is wrapped around the nucleosome. So what is going to happen is that the DNA is going to become more condensed. So if this long region, okay, this long region is now wrapped around, you can immediately see that this part has now become only this much. Okay, so it is becoming condensed. So that part of the DNA which winds around the nucleosome is what is called as a core DNA, and this core DNA makes almost 14 contacts with the host histone octamer. The region of the DNA which is between two core DNA, that means between two nucleosome units, that DNA is what is called as linker DNA. And this, to understand what is the number of base pairs of DNA that wraps around the nucleosome, a study was carried out with micrococcus nuclease. As the word suggests, nucleases are enzymes that will cleave DNA, but it will cleave DNA only if the DNA is accessible. So therefore, the understanding here is that the linker DNA, which is not bound to any protein, will be accessible to nuclease, but the wound DNA wrapped around the nucleosome will not be accessible to nuclease, and so the wound DNA will remain intact while the linker DNA will be cut into individual nucleotides. So with that, what was observed is that every nucleosome is wrapped around by approximately 200 base pair, but when the entire accessible DNA was cut, it was found that it is about 146 to 147 base pair of DNA that is wrapped around one nucleosome. Now, the wrapping around, the twist that the DNA makes is considered to be left-handed supercoils, and it makes an approximate of 1.65 turns, and the DNA enters the nucleosome and exits the nucleosome at the same point. This is very interesting. So it will enter from here, it will wrap around, and then it will come out at exactly the same position. That is something that has been interesting to be observed. Another important fact that has been observed is this, that the dimension of the nucleosome is 11 nanometers, and that is why it is also called as the 11 nanometer fiber. The linker DNA can be anywhere between 10 to 80 base pairs. And because the DNA has been packed to form a bead in a string structure, beads in a string structure, the packing ratio is 7. What is the packing ratio? The length of the unpackaged DNA to the length of the packaged DNA is what is going to give the packing ratio. So by just forming the chromatin, you can say that the DNA has been condensed by a factor of about 7. Okay. So you can already see that about 10 times the DNA has got condensed. Now we just look at the structure of the nucleosome in a jiffy. The nucleosome is made up of 4 different basic histones, H3, H4, H2A, H2B. So H3, H4 form a dimer, and then you have 2 dimers coming together to form an H3, H4 tetramer. Then to this tetramer comes and binds H2A, H2B dimer, that is 2 H2A, H2B dimers. So now you have 2 units each of H2H2B and 2 units each of H3H4 to make an octamer. And the DNA winds around this octamer that is there, and this nucleosome DNA complex is stable. That is why the chromatin structure is quite stable. Then the next level of condensation, see, you must understand this is only the first level of condensation. For the next level of condensation, you have another histone that binds at the site where you have the DNA entering one nucleosome and exiting the nucleosome. So when it binds, it's almost like tying it's almost like having a tie around a collar. Okay, so every nucleosome therefore is packed further, and you can see how you have now nucleosomes forming a zigzag structure like this. So this will continue, you see, a zigzag structure. So now you can see that where you had another nucleosome present over here, now you have the nucleosome present over here. So again you have got the structure becoming more condensed than earlier. So this condensed form is what is called as a 30 nanometer fiber. But this is only the second level of condensation. The final level of condensation is definitely the chromosome. And we all know that the chromosome is formed at the M phase of the cell cycle. So we know that the cell has a cell cycle. So the cell is present as interphase and mitosis and the M phase. In the M phase, you have the cell actually undergoing mitosis. So you have the different stages of mitosis: prophase, pre-metaphase, metaphase, anaphase, telophase, and then cytokinesis. So during the entire M phase, the DNA is highly condensed. So that is called as the metaphase scaffold chromosome. So you can see over here how a looped DNA is further looped to form a more condensed structure. And this looping is carried out by a set of proteins which is called as condensins. Now, please to understand that we've said that if a cell has to undergo cell division, then it has to have its DNA replicate. Why? Because mitosis is a simple understanding that if the mother cell is 2n, the two daughter cells that you would get is also 2n, which means that if the two daughter cells have to be 2n, then the mother cell has to become 4n from 2n. So 2n to 4n is basic replication, duplication. And that replication happens in the cell. So you can see over here how the sister chromatids after replication that are formed, they are called as sister chromatids because they are the same copies. They are the copies of the same DNA. So they are held together by a set of proteins which are called as cohesins. So all sister chromatids are bound together by cohesins. Now the question here is, why do you need to bind the sister chromatids together? Because in mitosis, when the sister chromatids have to be pulled apart, if they are not condensed and you pull it, then because they are tangled, they may break. But if they are condensed, you are minimizing the tangling, and therefore you are minimizing the breakage of the DNA itself. Therefore, condensation of DNA is extremely important when you have to have mitosis to occur. So this is a process which is showing that you have the sister chromatids further condensing to form the chromosomes that are present in the metaphase stage. And very clear indication over here is that a sister chromatid may have genes that can be expressed, but when it is highly condensed and in the chromosome form, none of the genes are going to get expressed because they are highly condensed. Okay. So effectively what I was trying to say is that gene expression can be controlled depending on how much the DNA is condensed. Well, people say that nucleus is an organized structure, and it is true. Why do you see that? There are several instances of studies that show that nucleus is highly organized. This picture that you see over here is basically showing that every chromosome of the nucleus has a space of its own, which means that the nucleus is compartmentalized and every compartment has a chromosome present. And we know that chromosomes are bound to the nuclear lamina, so they have a hold on the nuclear lamina and then they are positioned in one particular place within the nucleus. So as you can see, all these are different chromosomes stained by different fluorescent tags, and you can see that very distinctively every chromosome has its own place where it is residing. So if chromosome 1, this is the territory, and chromosome 2, this is the territory, there is no rule suggesting that DNA from these two territories do not meet. They can meet, okay, and where they are meeting, you can have genes getting expressed for a particular role. So two different genes on two different chromosomes can be expressed at the same time because the requirement is so. So that is another thing. Regions of different DNA can come together for genes to be transcribed, where it becomes dense as machinery for transcription and post-transcription modifications. They come together. This is a picture where it shows how processing is also compartmentalized. You see, it is not dispersed, but in certain regions, you can see that it is highly focused. So you can see that these are the regions where mRNA processing is happening. And so it is said that again, it is compartmentalized. So these are, there are many other studies that are showing that within the nucleus, things are very highly organized. Okay. Now we come to nucleolus. So nucleolus is a dense region within the nucleus, and one can see that there are certain regions that are called as nucleolar organizing regions. Within this nucleolar organizing regions, what is observed is that a lot of RNA are being formed, and proteins bind to the RNA to form what is called as the small subunit of the ribosome and the large subunit of the ribosome. So all the proteins that are required for forming the small and the large subunit, they enter through the nuclear pore complex, they get assembled at the nucleolus, and then they are exported out so that translation can happen in the cytoplasm in the eukaryotes. So the nucleolus is a region where you have assembly of the ribosomes happening. So there you have a high transcription of ribosomal RNA, okay, and you have a lot of proteins that are required to form ribonucleoproteins to form the small subunit and the large subunit, and once they are formed, they are exported out of the nucleus. This is a picture showing that just as you have cytoskeleton in the cytoplasm, in the nucleus, within the nucleus, you have again a network of proteins which form what is called as the nuclear matrix. And you see, it is this proteins that become the support for an RNA to bind or DNA to bind. So these organized thing that is there in the nucleus to a great extent is thanks to the nuclear matrix that is present. So this is something that is an integral part of the nucleus. If you remove all the protein, if you remove all the DNA, etc. from the nucleus, you will find this meshwork of proteins present within the nucleus, and that is what is called as a nuclear matrix. So let us make the conclusions. Nucleus houses the hereditary material, with the outer nuclear membrane and the inner nuclear membrane being a protective barrier and yet to connect with the outside. The outer nuclear membrane and the inner nuclear membrane fuse at regions where the nuclear pore complexes are enabling traffic across the membrane in a regulated manner. An important facet of the nucleus is that it is able to condense DNA to accommodate these in an available space, as well as make it a basis of regulation of gene expression. Transcription, post-transcription modifications, assembly of ribosome units, compartmentalization are features of nucleus that make it a very highly active organelle. So the nucleus comprising the genetic material is an active unit that requires constant transport of molecules between cytoplasm and nucleus, and a regulated gene expression at different stages of the cell, deciding what the cell has to do, and therefore it is called as the administrative unit of the cell. Thank you.